Põe lá.
É sexta de manhã.
Sexta de manhã? Sexta de manhã do aula. Não dá.
Ah, people. Stephen just told me that there was no sound. So starting all over again.
Take two.
I'm not the best Steven Spielberg. Okay, I'm a scientist. I'm not a director. So let's start again. Hi, everyone. It's very nice to see you again here on Thursday, on Friday. This is the second day of our meeting, and it's our first English talk in the symposium. And it's my pleasure to receive here on the screen, of course, not on my floor, Stephen Ekker, Professor Stephen Ekker. I'm going to introduce him. He has a bachelor in genetics and developmental biology in the University of Illinois. Also electrical engineering, a bachelor in electrical engineering in University of Illinois. He has a doctor of philosophy in Johns Hopkins University, postdoctoral fellowship in University of Washington, and again in Johns Hopkins School of Medicine. Well, I'm gonna just say this part, Stephen, because you have a lot of stuff to talk about. But it's all, again, guys, it's my pleasure to have him again. I hope you really enjoy his talk. It's amazing to have this opportunity to listen to Stephen, Professor Stephen Ekker. And I'm not going to say the floor is yours because there's no floor, but the screen is yours.
So the electrons are mine. All right.
Thank you.
All right. Well, thank you. Okay. Let's see here. Good. Are we good?
Yeah, perfect.
Okay, awesome. Thank you. Well, it's a real pleasure to be here and chat about my science. Thank you for the organizers, and thank you for attending. So the title of my talk is focused around how my lab has been using zebrafish primarily to pioneer work in gene editing and genome engineering. And I want to start by really thanking my team. This is a recent lab shot in the middle of a downward trend when we had a gap between our COVID this summer. We were outside in a beautiful arboretum, and I want to highlight a few key scientists. I want to highlight Dr. Ankit Sabharwal, who's going to I'll end with on the mitochondrial work, and then another set of scientists, including Gabriel Martinez Galvez, Hiro Ata, and Santiago. I'll highlight those as we go along. In addition, my colleagues, Dr. Carl Clark at the Mayo Clinic, and then the colleagues at the Iowa State University, including Dr. Jeff Essner and Maura McGrail, and Dr. Weirson and Jordan Walker. All right, I'm going to start with, what are we talking about? We're talking about, I'm going to use the words precision genome engineering. And in this case, I mean to the nucleotide level precise. So for example, here are some of our 3 billion base pairs of human DNA. Here's a sequence including that red A, and you want to be able to make a change, for example, to a T, or you need to be able to make a small deletion or insertion, an indel, or this, as I said, the substitution. These are all precise. We're not talking, and today I'm focusing on the ability to make these kinds of changes to the nucleotide level. We can do lots of other scenarios, but that's the core. And my lab does this with the logical component I call triangulation. So we do a lot of work in human cells in vitro and then zebrafish embryo in vivo. And we look for opportunities where the same science applies in those situations, with the idea that that's likely to be also true in us. And this idea takes advantage of the evolutionary distance between fish and humans. So being further away is more valuable with this kind of an approach than looking at a highly related organism. My lab has a core value of using these technologies, for example, to make disease avatars. Take human patients, they'll come to Mayo Clinic with a rare or undiagnosed disease, we will actually help build out models. It could be in zebrafish, and by targeting the change in the orthologous gene, and the goal is to understand the biology and ideally coming up with a treatment option. And we also can do this in a dish. So it's disease in a dish or disease in a fish, and those models are our core. So I'm going to talk a little bit about that strategy today. So the learning objectives, I thought I would talk about four precision gene editing methods that are coming online. One is an update, and a couple are recently published or about to be published. They all have bioRxiv preprints available if they're about to be published. But the point is these are going to be available to you. And so I want you to be exposed to them. And the goal is that you understand them well enough if you wanted to use them in your own laboratory, so with your own science. So I'll give you some examples, but really that's the goal. I'll remind you again, you can contact me at any point, either email or I do follow science Twitter. Some people ask me, we just have to communicate that way too. Okay, so the first story is GeneWeld. This has been out for a little while. I just want to highlight what GeneWeld does. GeneWeld is a way to target large cargo DNAs. And what we do in this with GeneWeld is we take a cargo and we put 24 to 48 nucleotides of sequence homology around the cargo, plus a sequence that we call the universal guide sequence. So it's a sequence that is not found in either the zebrafish or human genomes. That sequence then is a universal guide that we have that works really well. And then we have a genomic sequence that has a guide sequence that you check. So the only guide you need to design is the one site in the genome you want to knock in. And then you build this cargo out with those sequences. And this works as follows. So you have, if you're targeting in knocking into exon 2 here of this theoretical gene, you add the sequences to the left and to the right around your cargo. If it's a GFP transgene or something else you want to do, then the cell will resect the ends. You will have then recombinogenic sequences that will then work on and include the the GeneWeld sequence, and then gets integrated and repaired. And we actually discovered this by accident. A group at Iowa State colleagues were working on this, and this is just some data around the length of the ends. And this was surprising because all of the data in textbooks says that the longer the ends, the higher the integration rate. And that's true for homologous recombination. This is not homologous recombination. This is using likely a single-stranded annealing targeting sequence or microhomology mediated end joining. I'll talk about that in a minute. But what's really diagnostic around this is that you have this peak around 48 nucleotides, or depends upon what you're doing for 24 nucleotides, and it's more efficient. And in fact, when you do the comparison, so these are published, it's at least 10-fold higher than traditional homologous recombination. All right, so this is one of the early experiments that was focusing on the floating head gene, and you ended up having this beautiful notochord target expression with a red fluorescent protein. This was one of the early work. And this was just an injected animal. This isn't germline. And we realized that this tool was likely to be hugely valuable for zebrafish. The initial paper was published ultimately in eLife last year. And we just came out with a bioprotocol paper, which is here. And my thought was, if you have any questions on how to use this technology, grab the bioprotocol. We're really trying, the purpose of this was really to try to make this technology amenable to any lab that wants to use it. There's a series of vectors that are also out, so you don't have to start from scratch if you want to tag your own gene. So I'm just confirming everything good on audio.
Perfect, Ekker. Yeah, it's working fine.
All right, great. So that's a gene weld. Just wanted to bring people up to date. I want to move on then to thinking about screening, F0 screening work in particular, or if you just want to be able to reproducibly make a frame shift allele in your gene. And this work, I'm going to tell you, was started and was done by now graduates, Dr. Hiroada, Dr. Gabriel Martinez Galvez, and was actually made by, it was also an accidental discovery when we were doing small non-homologous end joining, trying to do non-homologous end joining based frame shifts. And we actually were finding some interesting, unusual, and unexpected science, and again, in collaboration with Iowa State. Okay, so this diagram, which we put out a few years ago, but it's really to get at this idea. If you are trying to make and use standard CRISPR systems, for example, and you put a double-stranded break, you will get a huge array of outcomes. Really, every cell that you add a CRISPR system to will give you potentially a different outcome. And they will often, they're represented here by these cartoons of different scenarios. You get heterogeneous molecular outcomes. That's the key. This is very complicated. If we try to do this in your fish system, some of those are going to be frame shifts and some are not. And some of those not frame shifts might not even be loss of function. It's very complicated. So you often have to screen through, you often have to then isolate and then clonally expand. It's not that it doesn't work. It's just this is a complexity issue around the fact that non-homologous end joining gives you quite diverse outcomes. And so this is often how crispins are used, although some of the works that now where you make deletions, where you use dual guides, is a little better because you're really taking out a segment of a gene. Well, what we had rediscovered was that there's another pathway that is often activated, and it's called microhomology mediated end joining. And what's exciting to us is that you get a same or very similar outcomes when you introduce this process. And that gives you less heterogeneity in your outcome, genetic heterogeneity. And it makes possible some screening and some biology that is very difficult with the randomness of non-homologous end joining. And thanks to our reviewers, they labeled this PRIMA alleles, which stands for predominant microhomology alleles. And we define that as situations where 50% or more of the outcomes, the edited outcomes, are typically one allele. And so those are PRIMA alleles. And some of this is out there, but I'm going to talk about an update here. And just reminding you what that is. So microhomology mediated end joining is local homology. And I'm going to define that as 3 to 10 base pairs of sequence. So it's the same sequence around a double-stranded break. It doesn't have to be contiguous. There could be gaps between those. But the result is a little bit of exonuclease trims it out, and the cell will anneal and heal. And sometimes I call it self-healing. It's a process because you don't need even the cell to help you find those together, because the homology will find the ends. And this can be, when it's true, the predominant pathway, the most common pathway. So it will happen preferentially over an HEJ. And the question has been, when does that happen? And can you find the rules for that? And this works great in zebrafish. Okay, so in fact, we rediscovered this in zebrafish, and everything I'm going to tell you about is that triangulation approach. We started in this case in zebrafish, and then we went back to figure out what was going on in mammalian systems. And so here's an example fish locus. We targeted tyrosinase to make albino fish, and this is a PRIMA allele where 88% of the alleles that we were generating with this guide yielded identical 4 base pair deletions, CTCA deletions. And when it's a high efficiency cutter, you can also then use that to generate your F0 animals with phenotypes. This is the tyrosinase phenotype, nice tyrosinase phenotype. In this case, nearly 60% of the fish are essentially nulls, genetic nulls, from the injected animals. So we published an initial algorithm for this called Menthu around understanding the kinds of sequences and the gap and for this. And that was exciting. That came from about 50 different zebrafish loci. And it has some understanding of mechanism. There was a lot of other work going on in parallel in mammalian cells, and using primary cells, in this case, embryonic stem cells, it turns out that the repair pathway for those cells are closest to zebrafish. So cancer cells in the lab, like 293, are not really zebrafish. There's actually a lot of differences in repair pathways between 293 cells and zebrafish in vivo, so certainly zebrafish embryo. But the mouse embryonic stem cells and the zebrafish embryo was the closest environment. And so as a part of this, we did a data set with 5,000 cutting sites. So 100x in terms of the number of sequences to probe. And we did a series of analyses, including machine learning, to understand that. And we looked at the Menthu scenarios, and Menthu was working quite well, and was actually, there's several other groups, 3 other groups were working on this. And the Menthu algorithm was as accurate or more accurate as all the others for making deletions and predicting deletions. So in reality, if you start with a sequence with this algorithm, what do you get? You'll find about half of the potential sites that actually exist, and you're right about half of the time. So this allows you to think about this from your science. My recommendation is design 2 or 3 guides if you want to make one of these alleles, and most of the time you're going to get a good cutter with a prima allele outcome. And that was true, and that's an update and in a scenario. We've then gone on to use and integrate a second MMEJ-based algorithm that was published using pure machine learning called Lindell. And Lindell identifies another subset of predictable outcome changes, which is a single base insert after SB-Cas9. We actually don't know how this works. This is actually quite of a mystery. So this is not a hypothesis-based scenario, but Lindell will accurately find single base inserts. And those happen in zebrafish at some level, and they do happen in mammalian systems. And so what we did was we updated our algorithm system to make it Menthu plus Lindell to make something called Mendel, easy for the geneticist to remember. And this increased our possibility of finding these kind of prima alleles where you want to induce a frame shift. So one base insert, likely to be a frame shift if it's inside an open reading frame, or as in addition to the small deletions. And this came out a few months ago in NAR, and the algorithm gives you a higher likelihood of making and predicting a frame shift allele from your sequence, which is often what we're trying to do for loss of function in protein coding genes. It facilitates these kinds of F0 screens where you inject a CRISPR system and you get many cells homozygous mutant for your locus. And this has the advantage of working much later in time than morpholinos, which often have a challenge, especially getting to day 4, day 5 screens. I have a colleague at the Mayo Clinic that has successfully used this to do adult genetic screening using this approach. It's very exciting science. This also lets you code exact same allele in your germline. So you grow the F0s up, you just raise some of them up, and then you screen. And because many of the sequence alleles are the same, you just double check by sequence and you'll get your F1, and then you just outcross the F1, and then you can expand for your F2. So you end up having highly reproducible genetic changes. There are opportunities to express these kinds of guides together with maybe tissue-specific Cas9 for tissue-specific mutagenesis, again, because you're knowing what you're getting in terms of genetic outcomes. And then the last one is interesting. I didn't expect this to be so important, but it's hard now. It's getting even harder to ship fish across international borders. The craziest one is between the United States and Canada. You don't normally think of that as a challenge, but it's very difficult for, I'm in upper, I'm literally a state that abuts Canada, and I can't ship my zebrafish to labs in Toronto, for example. And so the ability to just share this or here, use this guide sequence, you will generate the exact same allele in your genetic background, has been really useful. So I didn't think that this was the huge value for why you would do an MMEJ based allele, but it turns out to have interesting values from that point of view. All right, so those two are really represent an update of technologies out there, and some of you have seen these or you've had the ability to see them. I want to talk about two stories that are really new, and the first is focusing on small base changes, and one is to make changes in the nuclear genome, and then one is to make changes in the mitochondrial genome. And just from a human genetics point of view, this is a slide from David Liu's lab. Almost 90% of known pathogenic human genetic variants are from small genetic alterations. So there's a little bit of a bias here because we've looked for these kinds of genetic changes and we found them. There are likely to be others we haven't looked for, so I haven't found, but still a large fraction of our genetic diseases are from small genetic changes, all right? So that means we need tools. If we ever want to do gene therapy, true gene therapy at this kind of scenario, we need to have tools. And this work was also started by Hiro Ada when he was as a side project as an idea in his lab before he's an MD PhD student. He's now in residency, and was picked up by Brandon Simone, who defends on Monday. So he'll be finishing up. And as you know, I don't have to preach to this audience, right? Using tools like single-stranded oligonucleotides and HDR, homology-directed repair, is relatively inefficient in the larvae. I just want to offer that the technology I'm about to show you is the first tool that seems to increase this in my hands in a decade. So we characterized, about 10 years ago, the first germline HDR in zebrafish, and I've spent a long time trying to find improvements, and this was the one that seems to work well, and it seems to be universal, meaning it works in human cells and it works in zebrafish. Okay, so here's the challenge. If anybody is trying to make a single nucleotide change or a small change, when you add, this was actually from a 2018 paper where they actually quantified these errors. In our experience, this is the best outcome. Getting 4% or so of HDR of any sort of knock-in in a locus is really good in fish. It's just the efficient. Often it's 1% or less. Okay, but it's not just the overall knock-in rate. It's also the rate of issues of 5' sequence scarring, where you end up don't have the sequence outcomes that you want. And so this issue has been a challenge. One strategy that a group has published on is the idea that you localize the donor DNA molecule to the site of where you are introducing the double-stranded break. And this group, which is actually from Dr. Gordon's lab at the University of Minnesota, actually made a special Cas9 protein to do this, where you tether the donor molecule to the Cas9. And they showed that when you did this, you got an increased in efficiency, 2.5-fold efficiency in human cells. And that was interesting to us because we're at a marginal success rate in zebrafish, and we were shooting for even a 2-fold rate increase could be helpful. And so we thought we could try this, but we were wondering whether there wasn't a simpler way to do this. So rather than trying to use a special Cas9 system and a protein that actually was not commercially available even, could you come up with a strategy where you could use the current techniques but adapt them in a simpler way? And that's what we did. And that's called CRISPR Donor Guide. And the donor guide actually describes the method. Okay, so the idea in this case is that we use the dual RNA CRISPR system. So the CRRNA and the tracer RNA are separate, and that's for synthesis issues. And in this case, we add a tail onto the RNA with single-stranded DNA. And we often do this with simply as simple as 12 nucleotides of homology around where you want to make the change. So these, the red nucleotides flank the cut site on where the guide RNA or where the CRISPR system will cut. So that you are introducing the HDR, except the donor molecule is found in the CRISPR system, the RNP that you inject into the fish embryo. And again, all the strategy has been done is to develop and test this where we use, in this case, RNPs in human cells. We just use 293 cells typically. And then we deployed them in zebrafish for microinjection. And I'm just going to show you a few examples. So here's an example where we are using single nucleate or specific nucleotide changes, actually 4 nucleotides in one little cluster, where you're going to change the green fluorescence of the reporter to blue fluorescence. And we happen to have this exact same construct in a transgenic zebrafish as well as in transgenic human cells. And the reason I like this experiment is that the only outcome you're going to get of the blue fluorescence is when you have made the identical sequences and left everything intact. And you're also targeting an expressed locus. You're not just random sequences. So this is really mimicking, if you're going to try to make a small nucleotide change in a protein, that you want to do for modeling human disease, you're really focusing on this exact scenario. So you're going from green fluorescence to blue. And what we did with this outcome was do this kind of experiment. So here's an example where we are changing, there's a 6 nucleotide sequence where you're changing the precise nucleotide changes to go from this the 60 amino acid 65 and amino acid 66 a spot in GFP from a TY to an SH. Okay, so we have to make those changes, precise changes. And we looked at the nucleotide sequences for precise homology-directed repair. And when you do single-stranded oligos with this, we're getting, maybe 1%. Actually, it's less than 1% because we're ignoring all of this 5' scarring. It's about 0.5% at peak. When we add the donor guide, we're getting up to and including embryos with over 3%. So this was actually really substantive increase. So this was our first scenario. We do the same experiment in vitro in human cells, and we use cell sorting to get at, and we get a very similar ratio increase. Again, where we see a nice jump in overall accuracy and overall knock-in rate in human cells. Now, it is interesting that 293 cells do not have the 5' scarring issues in zebrafish. We actually get a net from a precision, a higher overall increase rate in donor guide in zebrafish than we do in human cells in the 293 cells. We tried a little bit in the equivalent of ES cells or IPS cells and don't quite see it all, but we definitely see the overall jump and increase. This approach is also true in terms of using gene editors to tether the molecule, whether you use a non-CRISPR system. In this case, we used a TALEN to introduce a double-stranded break, and we used a so-called dead Cas9 to tether the donor right around where the TALEN system would cut. And in this case, we also see a very nice 3-fold or more increase in overall precise homology-directed repair, suggesting that this really is working by tethering, right, localizing the donor around the cut site. And I think it makes sense when you're introducing your double-stranded break, the donor is there ready to be incorporated at the same time. So you're not having to compete with other processes like non-homologous end joining or microhomology mediated end joining that are going to be most common. And so what do we know about this? We know it works in multiple cell types in vitro. We know that this reduces that 5' sequence scarring rate in vivo, which may be a different mechanism. We have some ideas on that. Those are still open. It's effective as a two-component system, which means that in reality, we think this should be applicable to any gene editor. Maybe there'll be some new gene editors down the road. And the core methodology is in our bioRxiv preprint, and then the page proofs are all through. It'll be online with the official version any day now in the CRISPR journal. And so happy to talk about that. So the last module, I think we're okay on time, is to talk about something we've been working on in my lab nearly a decade, and it's trying to understand something I call the intracellular microbiome. So our mitochondria. Our mitochondria are amazing organelle. And they're not just the powerhouse of the cell. And in fact, that hypothesis has clearly been invalidated. It's invalidated by all the human patients that have mitochondrial disease, and they have a huge array of different phenotypes. And if it was just the powerhouse of the cell, you would be able to just look at energy use and you'd be able to figure out what's going on in the patient. It's just not the case. And the sad part, I'm at the Mayo Clinic, is that we see these patients and there are few treatments and there are no cures. We have many patients that come and they just really don't have any hope for what they're doing. So we've been focusing in my lab to think about the fact that there's actually many patients. There's some common diseases, including Alzheimer's, that are likely to have a major mitochondrial component. We know Parkinson's does. Mitochondria are the only organelle in animals that they have their own chromosome. They have their own genome. There's 37 genes coded in mitochondrial DNA, perfect conservation between human and fish. And for those that don't think about this, each one of our cells can have hundreds of mitochondria, and each mitochondria can have many copies, such that on average, some people will cite 8,000 mitochondrial DNA copies per typical cell. So it's an interesting question. It's quite different biology. And some people call this a population genetics problem. Again, I like to think of it as an intracellular microbiome. We'll get to it in a minute. So this last module, I'm going to talk about science led by Dr. Ankit Sabharwal in my group. He came to my group after doing graduate work on studying mitochondrial disease and science in the IGIB in Delhi, India, and has really been focusing on this problem leading a wonderful group and collaborators. And this work is also in press. There's also a bioRxiv that has all but the mitochondrial modeling, disease modeling science. I'll talk about that in a minute. And as a genome engineer, what are the challenges? So first off, everything I told you about non-homologous end joining and microhomology mediated end joining, none of that is true in the mitochondria as far as we can tell. No, nor is homologous recombination. So it's a real problem if you want to do it. You actually have this weird scenario of all of these copies of mitochondria. There's actually examples where genomes get transferred between cells. It's very complex. And most critically, we actually don't have any effective way to get RNA or DNA into mitochondria right now. So almost everything right now that's being done in the field is with protein-based import, because you have to cross the plasma membrane, then you have to cross the outer mitochondrial membrane, and then the inner mitochondrial membrane. And then, as I mentioned, all these mitochondrial copies, and those can also vary from tissue to tissue and with disease and age. So the real breakthrough in terms of the ability to make major changes was published by David Liu's lab when he collaborated with the Mugus lab out of University of Washington, where they had discovered a double-stranded DNA cytidine deaminase. And this allows you to make changes, basically a C to T change, when your target is a double-stranded DNA molecule. We've been searching for this, again, almost a decade. We were trying to build it. They found one of these in the wild. It was used by a bacterial system thought to be a toxin, and it's called DDA tox. And this allowed, for the first time, precise editing of mitochondrial DNA. It was very exciting. they did this by fusing with not CRISPR systems, because you can't get guide RNAs in, but using tails, so tail repeat proteins. And although it was very exciting, the challenge to that was it was complicated to use. In fact, designing a single sequence that you wanted to make change required at least 4 different protein combinations, and each one of those protein combinations was difficult to build because tails can be difficult to synthesize. And I actually argue that their efficiency in that Nature paper wasn't really that great. In fact, almost all of the work that they showed was 40% or less editing efficiency, which it's okay if you can then clonally expand, but it's hard to expand what you're doing in the mitochondria. You really need to be much more effective if you want to use this. So we've been developing a system to make single copy tails. We've been doing this for now a decade or more. And that allows you a single tube synthesis, so rapid assembly time. And we've explored whether our tail system can be deployed with their editor. And I'm going to show you that with that and some efficiency increases, we can actually get over 80% editing efficiency with single nucleotide precision in the mitochondria. It's pretty cool. And then we are now able to generate the first animal models of mitochondrial disease. So for those that don't know what tails are, so tails are protein-targeted gene editors, and they're like Legos. There's 33 to 35 amino acids, of which 2 amino acids are critical in targeting DNA. And what we did was we made a library of about 300 plasmids that are so-called trimers, and they can naturally go together using the Golden Gate synthesis in a single tube to make, we typically make 15 MERS, 15 RVDs assembles. And this allows you to do a single tube assembly. So what do they look like? A mitochondrial base editor is a 2-component system. You have a left half and a right half. And those 2 parts will come together in the middle and generate your sequence change here in the middle. And that change then leads to your outcome that's stable in the mitochondria. And so we were interested in whether the infrastructure we had been using for a decade would apply to this. And of course it does, because I'm talking to you about it. But before we started, we were interested in what can you do in our genome, our mitochondrial genome? What are the options that these tools enable? And so we started with an in silico prediction work, and this was done by a graduate student, Santiago Restrepo Castillo, who's actually an international student from Colombia at the Mayo Clinic, and in our virology and gene therapy program, because he's interested in trying to make gene therapy approaches for patients with mitochondrial disease. And what I've shown here are 2 circles. They are the 16,000 plus nucleotide circles of human and zebrafish mitochondrial genomes. They're very similar. It's amazing how similar they are in size. And all of those 37 genes are perfect synteny in human versus zebrafish. And what we've done here is mapped out the protein coding genes and then looked at with the red dots, or the dots, if you're can't see the red dots represent where it's likely to be a protein truncation. So you're likely to have truncated the protein. So a frame shift, or equivalent of a frame shift allele that most people use in CRISPR systems and nuclear genomes. So you're likely a candidate loss of function allele. And by using this algorithm and leveraging the requirements for TALENs for having a T and then the sequence changes, we were able to identify a software tool called TailWriter to predict. And he predicts that there's over 1,500 potential target gene editing sites in human mitochondrial DNA and zebrafish DNA, very similar, and about 100 of which are likely to induce a protein truncation, so a PTC allele. And we were then using those loci for everything that we're doing. And so the question has been, does the system that we built, the toolbox we built, does it work in mammalian cells comparable to what was done? And then can we deploy it in new areas in zebrafish? And that's what I'm talking about. So the first step is that we used and targeted the exact same nucleotide sequence that was published by David Liu's group, and in 293 cells. And we show that our system is indistinguishable in terms of efficacy. It's about 35 to 38% gene editing in the similar systems done in parallel with the same specificity. So that's really good for understanding what's going on in human cells, but can we do this in vivo? And more importantly, because I do a lot of fish work, can we do this in zebrafish? And to do the fish work, we had some homework we needed to do. We needed to be able to genotype the animals ideally without destroying them. And so we also developed a non-destructive mitochondrial DNA genotyping protocol. And I'll show you that in a minute. And that allows us to do functional validation and making novel mitochondrial disease models. So Ankit labels this FISH MD for finding innovative solutions for human mitochondrial disorders. It's a fun story, and it happens to be true. So here is our pipeline. So we take and inject the single cell zebrafish embryo with the base editor RNAs. We do the left and the right for your locus, and I'm going to talk about CO3 and TL1 loci, a protein coding locus and an RNA encoding locus. Then we raise them until day 3, and at day 3 we put the fish in with a low dose of proteinase K to get enough cells off of them to be able to genotype, do just a simple PCR genotyping and or Sanger sequencing to confirm the fish and the percentage of the edits. And then those fish are then ready for longitudinal studies. We can study them directly at typically day 6, day 7, day 8, or we can raise them up for germline transmission or other outputs. And this has been really rapid. We've been able to do really substantive work in just a few months once you have the initial system. So everything I'm going to tell you about functionally is from F0 animals that were de novo regenerated in terms of the gene edits. And so here's some examples. So here's the CO3 locus, protein coding locus. And what was amazing to us right away was we were getting up to 89% editing efficiency from this locus. And it was really remarkable and with high precision. We can see that by using RIFLEP analysis. So we have a restriction site that gets lost. Or we can sequence these next-gen sequencing and show and quantitate and show that the animals that we are hitting, in this case, was 88% editing efficiency. This is consistent from embryo to embryo. For example, here are 7 embryos, of which 6 of the 7 are well over 80% editing efficiency. And in the case of single nucleotide, we were seeing very little off targets anywhere else in the mitochondrial genome. So that locus got us really convinced that the system has huge, enormous potential. We just need to be able to do it where we have nearly 80% editing efficiency. This is from a single dosing of those RNAs at the 1 cell stage. And this particular change is likely to be loss of function because of that 260 amino acid protein. We've cut off 100 amino acids, so it's only 160 amino acid protein left behind. And as I mentioned, it's precise. So this is sort of that ideal strategy of precision editing, single nucleotide editing, going in and removing a protein function. Okay, so that was exciting. And I'll show you, I'm going to show you molecular and biochemical readouts of these fish in a minute. But we are also interested in tRNA genes. And that's because pathogenic variants in tRNA genes are a huge fraction of the mitochondrial inducing variants in human patients, such as MELAS. This is a classic mitochondrial disease. And in the case of this particular TL1 gene, there's actually at least 5 known changes that induce it. Number one is a very common variant, but there's other variants in this tRNA that are likely to cause MELAS or associated with MELAS. And at the moment, we can't make, because of the limitations of the editor, that's a TC sequence that you need to edit, we can't induce MELAS de novo. But The human and fish tRNA, TL1, are very similar, especially when you load them up in secondary structure. And what Ankit did was he looked at these other MELAS variants and said, can I make essentially a MELAS mimic? Can I target the same spot in the tRNA in the zebrafish for making a potential MELAS? And that's what he did. And in this case, we're targeting the TL1 gene. And again, very high efficiency, well over 80% in many of our embryos. And you can see that by restriction fragment like polymorphism or next-gen sequencing and confirming that. And in this case, you also get embryo-to-embryo consistency in these scenarios. So you have a very high frequency making these changes in that loop structure of the TL1. And so giving us a really interesting tool. So the question is this actually MELAS? So I'll just tell you what first stages of this. So what's going on? So we collaborated with Dr. Eiko Oguso in the Children's Hospital of Philadelphia and the Marnie Falk's group, and just started doing systematic panels of mitochondrial function. And lactate is the most common. In fact, until recently, it was the only clinical test for many mitochondrial patients, including MELAS. And in the case of the fish, we actually have almost a threefold increase in lactate levels in those fish. And interestingly, the CO3 do not. So we were able to use these two mutants with differential specificity of the outcomes. We've done a series of other panels. I'm just going to show you some of the simpler ones around going after electron transport chain complex 1 or electron transport 2 or complex 4 or citrate synthase. These are just some classic mitochondrial metabolic functions. And what I want you to take away with is you see differential outcomes. So in the case of the CO3 mutants, complex 1, maybe it's a little bit compromised, but clearly complex 4 is much preferentially. And in the case of TL1, we're actually seeing both complex 1 and complex 4. And we're not seeing really anything on citrate synthase or complex 2. So we're starting to see really interesting hallmarks of mitochondrial disease. So elevated lactate in the MELAS-like variant, the TL1 mutants, and we're seeing this really specific decrease in complex 4 for CO3, and this more broader defect in TL1, okay? And at the levels of efficiency, we're not seeing any substantive ATP level changes, but that's actually consistent with known issues with mitochondrial copy number. We actually need to really suppress mitochondrial function especially in the 5-day, 6-day zebrafish embryo, really at higher levels if you want to really hammer ATP. That's in part because of all the maternal pools of mitochondrial function. So this last piece is that I just want you to, we've got and now hit to the point. This is the bioRxiv preprint, should be 2D barcode. If you're interested in this, we have a single tube synthesis allows us, with combination with Tailrider, to start thinking about editing the mitochondrial genome. The efficiency is we're able to take on and build and what's been published before, and really coming up with where you have new efficiencies, editing efficiencies of well above 80%. Gives us now the new stages for phenotypes. And the fish is, of course, a automatable in principle and high throughput toolbox. And in fact, we have some novel compounds that were being screened in human cells in vitro that we're going to use for testing for in vivo as a potential small molecule therapeutic application. And really, I want you to get across that my lab continues to use this rules precision gene editing through triangulation, again, using the human information plus the fish information together for really interesting areas. And I just want to share with the mitochondrial story where we're at. We started with people working in human cells. We deployed them fish in vivo, learned and built a system, in this case, highly effective RNA delivery system, updates to the delivery, and hit editing efficiencies you weren't seeing with mammalian cells. And we went back and used this approach to then look at in human cells, and in this case, primary human cells. So primary human cells, not just 293 cells. And this has been led by a postdoc in my lab, Dr. Babette Carr. And we're now seeing 75 and nearly 80% editing efficiency with high precision in the human ND4 locus. So we are excited by this because you can start thinking then If you've got the right combination of sequence variant in your patient as a patient, and can we start building out mitochondrial gene editors? Can you deliver those in vivo? You might be able to have the next generation of therapies. It's just an exciting state. This has only been possible in 2021. This was all new science. So I hope you understand where I'm at. My lab does a lot of this work. We focus on this idea of disease in a dish or disease in a fish, and we focus on that. So I want to end. If you're interested in any of these pieces, contact me. Often you can. I do check notifications on Twitter. I'm not in Twitter every day, but I will answer and collaborate. And then I've tried to make it that all of these protocols are available to you for your science, and always happy to help. And I want to thank you for your time, and I wish you well. I'm looking forward to hearing more of the talks for your meeting. So thank you.
Thank you, Ekker. Wait, trying to audition again. Thank you very much, Stephen, for the lecture. It was amazing. I can see like some people ask some questions here, and if you don't mind, can we ask you some questions?
Yeah, please do.
Is that okay? Perfect.
Yep.
So the first one, one of the people who watched asked, what is the possibility of inserting CRISPR in the zebrafish genome itself for the study of the treatment of genetic diseases, for example?
Right. So the issue is delivery. Our fish live in water and we don't. And so it's really about the fact that, if you're going to use a virus, you have to have a virus that will now infect both humans and fish, and viruses don't do this, right? I mean, so in many cases, the limitations of the triangulation is the delivery. So the products and the systems, once you've crossed the plasma membrane, most of the science is hugely relevant. It's just a matter of issue of now it's nanoparticles, and some of those things might be universal. And we've published a couple of papers on using nanoparticles, and we actually are going to be doing some nanoparticle science. So we'll probably test it, but then are you going to inject it in the blood? How are you actually going to get it into the fish? Anyway, just giving you a perspective. It's really about the delivery, the specialties of the delivery.
That's it. The first one. The second question is, how are these studies going for human genetic disease or HIV and the use of CRISPR in precision medicine?
So I ended at the end around, we're actually there are patients with mitochondrial disease with their sequence variant, and we are building a database. There's some of these have already been made. Dr. Eric Schoen in Columbia has done this. But these are the patients that actually walk in the door at Mayo Clinic. And I have a graduate student who is taking one of those patients, and we have made iPS cells. Mayo made iPS cells from those patients. So those cells carry their exact mitochondrial genome with their variant. And we're hoping to make a custom base editor to reverse their disease. And so this is really brand new. This is all 2021 science, not 2020 science. And we're really hoping that we can start making precision therapies for patients that literally have no options right now. It's a very exciting time. That's an example. There's many, many labs doing the work for the nuclear coded genomes. And there was a really nice paper on sickle cell disease by Beam Therapeutics, using base editor for sickle cell disease. And looks to me like they're on their way to making a sort of a universal gene therapy for sickle cell disease. That would be fantastic.
That would be fantastic, that's true. The other question is it possible to do tissue or cell-specific editing with this mitochondrial genome editing approach in zebrafish?
Yeah, so we want to do that. One, the challenge is expressing the two components. It's a left and a right arm, and they're pretty big proteins. We are hoping to make a single chain, so-called single chain system, where you can make a single transgene, and then that can be you could then put a GAL4 UAS. You make a UAS of that, and then you would just cross it into the GAL4 line, and then it would edit in those spaces. So it's on the roadmap. We have a couple of technologies we have to tweak. We have made a single chain TALEN for the first time that could do this for the nuclear genome. So I'm pretty comfortable that we have options, but there are there's details. It's going to be all about the details to make it work. But yes, that would be very exciting. Of course, there's really a New England Journal of Medicine article last year showing that if you are a patient with mitochondrial heteroplasmy, some of our cell lineages will actually naturally remove the heteroplasmy, and some will not. And the lineage that will apparently remove almost all the heteroplasmy and maintain wild type are our T cells. And so just looking in the blood lineages, you can have a huge, more than 100x differential heteroplasmy maintenance for B loss. And so it's very interesting. Let me say, it's going to be really interesting. Now, we can probably brute force it if we needed to by using a left and a right and making it dual transgenic. It's not impossible to think about that. But we were hoping to be able to do this with a single molecule version, because then that's much more universal for people.
Interesting. Yeah. One last question before we end. It's going to be my question. I'm going to take advantage of this. Since I'm doing the editing here and all the directing of video directing, I'm going to take this chance for myself. It's a general question, in fact. What would you say, what do you think it's the challenges, for example, for people who work in Brazil, for example, in the labs in Brazil, who has a little bit of limitations? I'm being nice. A little bit of limitations in money. Regarding funding. What would you suggest? Which way would you say would be the best way to aim gene editing if you don't have that much money?
Right. So almost all of the methods I showed you, accessibility is a key component. Prime editing is very difficult to do, for example, and although in principle I have nothing to object. I think it's really cool science. It's very complicated. Yeah. For many labs, the donor guide would serve the exact same output that what prime editing does, and have the exact same flexibility, essentially, and is 1,000 times cheaper, right? So I think that I think that almost all of the tools, we want to keep the cost down because we want to be able to do screens, right? Yeah. And so it's anything that's expensive, if it goes up 2x and I do 100 times, that's really a lot of money, right? So we have a motivation. I'm just sharing this. So, but reality is that Zebrafish Labs, I'll just share. I have a grant that's likely to be funded again for year 20, and it's the exact same budget as year 1, right? The exact same budget, right? There's been a little bit of inflation in 20 years, right? Yeah. So being able to make these tools accessible is a critical piece. So that's one side. I think that there are abilities to make proteins like CRISPR-Cas9 in-house, right? My lab's made a lot of reagents in-house, right? And I think that our next paper will include a method for making Cas12a, a really nice Cas12a for fish, and the key is that we make it ourselves. It's not commercially available. Yeah. And the cost is 10x cheaper than anything you can buy. And I think there's going to be, because a part of the innovation is that cost scenario. And so I think that making these tools accessible in practice, right, is going to be critical. And so if you're having any challenge, the guides, I still think the cheapest way to make the guides is yourself, right? Get an oligo order and then synthetic RNA, but you synthesize in vitro, right? It's a lot cheaper than a Synthigo design or whatever. But those are the key pieces. I'll just end with the donor guide limitations. Why are we only using 12 nucleotides or whatever? It could probably work longer. The goal was to keep the cost down because we wanted to be below the standard synthesis that IDT does. Yeah. So because it's one synthesis, it's just an RNA DNA, and they just switch out when they're doing solid phase. So it's not a lot more expensive than just making an RNA to do that donor or that chimera. If we had forced it to be longer, it suddenly shifts into this custom synthesis that's might be 10x more expensive. And so those were all in the design considerations. So I understand that. I concur. And then the hope is that if you make a good guide, sharing, right? So that we're all sharing. Obviously, it has to be the best way.
Yeah. Perfect. Thank you very much, Stephen. Thank you a lot for giving your time for us. It was a pleasure to meet you, and an amazing talk. Do you have any words for the people in Brazil during pandemic?
Well, I keep up the great science. It's all I can say. Science is how we're getting out of this, and it's really how we're going to make the world a better place. So thank you for all your hard work. And I am editor of the Zebrafish Journal. Feel free to send me papers. Always happy to see papers from Brazil, and always excited to see what's going on. And I'm hoping to plug into the YouTube channel and see some new science myself. So awesome. Thank you, everybody.
Perfect. Thank you. Thank you, Stephen. See you guys. See you in the next meeting.